Richa, Richa is a senior bioinformatics scientist with the Poly-enabled services team. After her PhD at IISc Bangalore, where she studied amino acid sequences and their relationships to protein structure and function, she went on to do her postdoctoral fellowship at IRB Barcelona. Richa has a deep background in structural and systems biology. She also holds significant expertise in the application of data science to understand high dimensional biological data. Just a couple of things to note before we begin. This webinar will be recorded and we will take questions in the last 10 minutes of the webinar. You can send in your questions in the chat window. We also have a support team here at your service. I would now like to invite Shwetab to start today's seminar.
Uh, Talking about some challenges with analyzing omics data and in general all kinds of biological data. This is a framework that we came up with. We call it the EPIC framework, and the idea is that all kind of biological data analysis face the same kind of challenges. So whether that be ingesting or taking up a large amount of data or a large amount of heterogeneous data, uh, or whether it be processing a large amount of data, as well as what we indicate in the figure here, there are many ways to do that, and therefore these processing ways keep changing, evolving very rapidly. Then we go to the stage where you have to interpret something, and it's not enough to get to a particular visualization, but you have to make sense of it in the context of the experiment, in the context of all that is known before, and finally collaborate. And all of these challenges we find are very common across different kind of data analysis, and as well as are faced by many different labs. Based on our experience over the last few years, these are very common and are a consistent account of this kind of data analysis. Why Shiny apps though? And as we talked about on the last slide, the challenges of collaboration as well as interpretation are enabled a lot by a framework like Shiny. Very often, I'm sure this audience understands, it is clear that there is a need for an interactive web application and interactive environment where data can be visualized and seen. And this need is increasing with the amount of data that is just being generated. And this on the slide you have a graph which is something that we use very commonly, which shows just the explosion in the amount of genomic data, and I'm sure this audience is very well aware of it. And with this explosion of data for interpretation, visualization, as well as collaboration, we need to be able to create web applications, which is again non-trivial if you're not using something like Shiny. This is something again that I believe that we can all agree with. And very often when reports or analysis are shared, they're shared in the form of static documents such as slide decks. The challenge with that is that there is a lot of back and forth. I'm sure all of us have been in a situation where somebody has come back to us saying that, okay, what is this particular dot on a volcano plot? And as frustrating as that is, you have to be able to answer that question and that builds a whole lag. Somebody responds with, "Can I understand this? What this particular node is?" And that requires a bunch of follow up back and through, and that can be avoided by using something like Shiny, an interactive framework like Shiny, where you can build a web application and send that to your team or your collaborators rather than a static document such as slide decks. Now we'll talk about some basics of what Shiny apps are and how they are made. Again, the idea is to give you a broad overview of the ecosystem. So Shiny is essentially a way to be able to write web applications, interactive web applications in R. And why R? Because R is a very commonly used language in this particular community. Bioconductor is the most widely used package in R. In fact, in almost all of the R world, and it's a common package that almost anybody who has done biological data analysis has used. So many people are familiar with using R, and by just using R, you can create web applications which are very professional as well. And we'll cover that later on, how you can use R and make professional-looking scalable web applications. this is an example of the kind of resources that are available with Shiny, and a good part of having an open source framework like Shiny, which is backed have a lot of examples to look at for inspiration and otherwise. And we list two of those here. One of another useful resource is Show Me Shiny, which contains links and descriptions of many Shiny apps developed across various disciplines. And you can look at them for ideas of how to show your data, how to visualize, how to present it. And very often the source code is available as well. There are many reusable widgets and interactive plots available as well, so you don't need to recreate them from the scratch, but can just use those widget by looking at the code base behind these kind of Shiny applications. This is a very basic template of what a Shiny app looks like, completely abstracted into just the bare minimum to show what that is. And in essence, there are only two parts. One is UI, which is the user interface. The other is the server. Server is what executes a request by the front end or by the user interface. So the user interface is used to describe the look and feel of the application. This is what you use to describe what the application should look like. And the server is where the actual work, let's say the data processing, takes place. And you have to define these two and then connect them into a Shiny app. What this final command where we say Shiny app UI is equal to UI says that we are defining a Shiny app and the UI is such and the server is such. And we just see by this very trivial example what how simple it can be. If you're using any other framework, it can usually include an understanding of many different components, but the community has reduced, in case of Shiny, this knowledge to two components, and by just defining them well, you can get your Shiny applications. Again, this is a trivial example, so as we mentioned below, as you get into more and more complex applications, you can split things into two different files or multiple different files, in fact, and that's what more advanced developers of Shiny apps do. But to just get started with something very simple is almost as simple as this, and this just goes to show how simple and powerful this framework can be for developing applications. Again, let us know if you have questions about these at any point. We are going to take them at the end of the webinar. This just lists the advantages that we have in some sense already discussed by now. We see a few different key advantages. The first one, I think, the biggest bubble as it shows the interactive display and data manipulation. This is not a static representation of data, hence you can interact with your data, you can play with it. You can also manipulate your data. So this is not a static representation in the sense that you can change parameters and see how your visualizations or how your analysis changes, and that's very easy to do. So interactivity, we think, is a big value add. The other is the ability for a biologist or a scientist to be able to be independent of other people or other teams for developing their own applications. And again, we believe that this kind of independence move faster. How do you share your app? And this brings us to the third advantage, which is collaboration. Once you have your application, you can share it with your team and collaborators pretty easily. You can either just share the code, which is what is represented in the leftmost tile. You can upload your code to GitHub or Dropbox and just share it. You can also host your own server, which is a way that more advanced users can go. And then there are ways to deploy it to the cloud without having to go into all of the complexity of managing the cloud infrastructure. Poly is one of them. We have mentioned shinyapps.io and RStudio Connect as well, and they do a pretty good job of doing the same. We believe Poly has that kind of infrastructure for this community, which can be very useful. Here, I'll hand over to Richa to take you further, and she'll show you an example of an application which is very relevant for this community. Richa.
The introduction. And now everybody has a fair good idea of what Shiny apps are and what we can do with it. But my part here, I would try to explain how Shiny apps have infiltrated into biological data analysis. So before we see how they have helped us in biological data analysis, it's important to see how the biological data is growing. So for now, if you're in the slide here, you can see that there is biological data analysis in midst of a revolution. And it's not just genomics data which is coming up. There are cheaper techniques, and because of that, you have more image analysis data as well. And even structural genomics consortiums have leading to a huge amount of data. So it's a very relevant problem for not just omics, but also other avenues where we need to have better tools. It's the need of the hour that we need tools that would manage the data, that would store the data, analyze the data, and hopefully help us interpret them better. So the key challenges broadly from all the other aspects of biology can be seen in this slide, that we need to identify the problems. But the major bottlenecks that we face in performing analysis, where we have different workflows, then we need a standardization for these workflows. The input from one needs to be standard for compatibilities. We also need to do a lot of quality control over the data that we have obtained. And another biggest challenge comes in how will you visualize this huge data in terabytes. And these require some mathematical tools like dimensionality reduction for us, so we can make sense of the information that we are having. So the biological community has been over the years developing tools to analyze the data. But here what we are going to discuss is how Shiny apps are becoming one of the easiest way to have biological data analysis. For most of the biological data that we have, we use R packages to manipulate or visualize or interpret them. And in this sense, integrating R with web applications or having a GUI for those makes it even more helpful for us. The biggest resource, yes, we have a lot of repositories and online galleries where you can search for each discipline and look for different types of Shiny apps. We also can go to publications, and we can see NCBI and the other journal websites. And I will now give you an overview of what different types of apps are there for biological community. Let's say more power to the biologist. Shiny apps are not just for data visualization and analysis, although it seems more like that. But we have seen by looking at the publications and the different Shiny apps that have come up that people are now using it even for data reproducibility. So you can submit a Shiny app along with your publication, and the readers and the reviewers can go through it using your Shiny app. Shiny app are also being developed as a portal to data mine and portal to analyze your various databases, which are very comprehensive, especially like cancer genomics databases, which have a lot of mutational information and other things. Similarly, Shiny apps now provide us with different GUIs, and it can aid in parameterization. So in the field of mathematical biology, we have a lot of predictive models where we have equations which will try to predict a behavior of a system. And these mathematical models often have a lot of parameters. So these parameters can be either first viewed, and also we can tweak around them. We can have a standard, finalize a standard for them as well. And also since we deal with a lot of different types of workflows, we often have more than one method to do a particular analysis. And using Shiny apps can help us explore and compare these different types of methods. Another important problem that we often face is from the data scientist or a bioinformatics point of view. So yes, biologists find it difficult to interpret and visualize the result, but even bioinformaticians also sometimes have problems in communicating the kind of analysis to the biologist. Also, over the years, with different iterations, people lose information. There are different iterations to the same workflow, and the history might be lost. And sometimes a lot of time is used in communicating with different teams to finally come up with an analysis or a workflow. So let's say there are a little bottlenecks in the software development cycle. So we plan and define a problem. And so after planning and defining, we have a data scientist who would analyze and design what needs to be done. And then there is a numerous iterations with the developer to find out what type of software we can use, what tweaks can be done to the software, what are the important features that need to be put onto it. And then we would have to, and then the development team takes it further to implementation and testing. So the major bottlenecks here is that a lot of time goes in communicating, effective communicating. And another important thing is that there is a long time from development to the deployment stage. However, if we have Shiny apps, data scientists that can develop them, it's a very easy or transferable information from a data scientist to a software. And another thing that Shiny would do that they seems easily deployable, it actually expedites this whole entire cycle, and we can simply deploy them after testing. At our front, at Elucidata itself, we have tried, Poly also forms a platform on which we can host Shiny apps other than the other platforms of sharing the app or through GitHub or sharing the app on shinyapps.org. So I would now move to giving you a demo of an app that we have just made for the demo purposes. And we have hosted this on Poly platform. And I would like, and this app, okay, a brief introduction about what we will be doing in this app is important. The app that we are going to show you is to visualize the downstream effect after an RNA-seq data analysis. So what is RNA-seq data analysis? The key buzzword is the transcriptomic analysis. So we have raw RNA-seq data that we obtain from either control cells or conditional cells. And these different samples, the raw RNA-seq data is then mapped onto a reference genome. And finally, we get an expression quantification where we can see that a gene A is expressed 3 times and the gene B would be expressed as 11. These are just numbers which tell you about the read counts. And once we have this expression quantification for different samples like condition 1, condition 2, and the control samples, we could run differential gene expression analysis in which we try to see what are the different genes which are upregulated and downregulated. But the whole platform, the pipeline that we follow usually is very, very tedious. And what we get would be just plots. And we would get a list of genes probably that are overexpressed or underexpressed. And this kind of analysis needs to be aided with something which is visual. And it's not just about being visual, but also being interactive. Because in a simple report that one shares, we often have to go back to see what genes are actually in, what are the expression values, and where does this gene lie in the actual plot. And if you were to just filter out the data for some specific genes, how would things change? So the app that I'm going to demo now would just give you how we can visualize these things, and also how an interactive interface helps us interpret better. So in the interest of time, I have preloaded so this is the app that we have for the demo. This is hosted on Poly, and it's called RNA-seq downstream analysis. It's a very simple, basic application that any data scientist can build. Here, what we have is a data frame, which is this data is being pre-processed. So we already have the full change values for which genes are upregulated and downregulated across 2 different condition and control samples. So here you can see that we have a control. 3 replicates, like 3 patients were treated as healthy control, and then we had 3 patients for condition 1, and then we had 3 more patients for condition 2. And once we submit this data, we would be able to analyze it. Just a second, it is just going to load the data and we would be able to analyze it. Okay, so here in the first tab, we were able to load the data that I just showed you, and then in the group plots we are able to see. So the first thing that we want to do is the 3 groups that we are studying: the control, the condition 1, and condition 2. Are they similar to each other, and can we draw a comparison between these groups? So in the first thing that one could do is to run a PCA map. Principal component analysis is a tool which would look at the gene expression across these different conditions and then try to find out which and try to plot them and try to see which conditions are clustered together. So here we can see that the 3 control replicates are similar, whereas condition here is a cluster of condition 1 and is another cluster of condition 2. We could also, the interactiveness comes into play that we have different principal components. So here we can take another principal component, say the 5th principal component, and then we could try to build the PCA plot for it. And this is the interactiveness that we are talking about. Similarly, we could also look at the sample distances, which is in the form of a heat map. And if we look at the distances calculated between these different groups, we see that condition 2, the condition 2 is a little more far away, or the expressions in we visualize in condition 2 are different from what we see in control and in condition 1. Okay. So the next part of the analysis would be that we look at the genes which have been upregulated or downregulated. So for this, we would look at we will try to generate a volcano plot. Here we can select the comparison between which groups that we want to do. The first one is the condition with the control, and the tool automatically loads volcano plots for different types of all pairwise comparison that one could have. This is here we see for the first. And we also, what Shiny app allows you is that you can now play around with, if you want to see the genes that are which are only changing for like more than 4-fold. So if we change the value here, it would change the plot as well. We don't see many of them which are up. This is a huge number. We would say 2. We could also change the p-value thresholds that we use. We could use a lesser stringent p-value, and here we could see the change in the plot that comes up. Another interesting feature for RShiny is the interactive nature of these plots. So here you can, if you just hover over the plot, you can actually see that these what this gene is about, and what is the fold change, what is the p-value that we are looking at. And if we really want, we have a very specific gene in mind, we could select that gene, and then the plot would be updated. And once the plot is getting updated, we would see these two selected genes here. So here you can see this gene that it's expressed, it's slowly expressed with not that much of a fold change. Okay. So moving on. From this, we would also like to see if what is the expression of a particular gene across the different conditions. So if we select a gene here, it will show you a box plot of the expression of that gene in different conditions. For example, here we chose a gene WDR78, and we see that in the control condition, this is highly expressed, whereas in the two other conditions, it's not expressed so much. Okay. With this, also enables us that now if we want to know what is the actual values or what is it, we could go to the data tab output folder, and so we would be able to download the information from that. Okay. Finally, for this app, we have the heat map. In the heat map, what we essentially want to do is we want to see the expression or values for the each genes, the normalized expression values for each gene across different conditions, and then we want to see if the different conditions actually clustered differently. So for the demo purpose, we will select, say, about 50 top genes based on the variation that we see, and you would generate the heat map. Yeah. So from the heat map, the first thing that we can see is that the condition 2 is a separate cluster where most of the genes are differentially regulated, and the condition and the control cluster we could see are similar in nature. And this heat map would give you a divide of what are the upregulated genes and the downregulated genes. And then these genes can be, we could have a folder in which the data is outputted, and we would download the list of genes which are upregulated or downregulated. And then these can be fed into the further analysis into enricher or other pathway generation. So another thing that we would show you here is that since this app is on Poly, we also have the Poly features integrated into the app. So Shiny app itself has a lot of features that you could do then without the knowledge of any other JavaScript or other things. We could have interactive features by using R, the power of R packages. And with this, we could, in Poly, be able to actually save the state. So if I click here, it would save a state, and then we would be able to save the entire analysis as a state 1. And later on, when we log into my Poly, if I log in again with the Poly platform and visit this same analysis, I could just restart from where I stopped. And the other key features that I would have on Poly would be that I could simply just share my analysis with others. So it is not just sharing the code or the input files. I would be able to share the entire project that I have, the analysis that I have. And the great part would be that every time another user is making changes, it gets saved as a different state. And then we could go back to we can go back and forth on different states that we have or the analysis that we do. Another thing is that we don't have to worry about the memory or how to access it, because there's dynamic resource allocation. And since it's on the cloud, we can access it from anywhere in the world. It also has implemented the GridT integration, which allows us to go back and forth on the analysis and the states. We also, for the Poly platform itself, there's a Python integration. And the biggest thing is if we host anything on Poly, we would also we can have a customer support anytime. Yeah. So with this, we would like to say that the biological data analysis is a Herculean amount of task, and it begins with data cleaning, manipulation, and at every step of the way, we have different tools that could help us visualize and interpret it better. But our Shiny has really played a major role in easing out the way for biologists and also for bioinformaticians, both. I would now like to thank everybody that was involved and also the entire team of Elucidata. We are a startup. We started about 4 years ago, but we are really growing. We have 60 plus employees. We have also published in many different publications and presented international conferences. And you can always visit our Elucidata website for further details. We have partnered with many different companies, including industry and academic collaborations. And our core expertise lies in metabolomics and big data analysis. So do look us up. Thank you. We would now take if we have any questions. Okay, so
It's an R server-based environment. So Poly is a general environment which can host code in almost any language. It can also take R libraries or Python packages or C++. But yeah, you do need to write Shiny applications in R, just as you would write Shiny otherwise. We provide certain plugins which can be used in your Shiny applications to make them hostable on Poly. In general, to write Shiny applications, you do need to know relatively basic R. One of the questions is how is this different from Metabo Analyst? I think the big difference is that Metabo Analyst is it's almost an end-to-end tool to do metabolomic data analysis, but you cannot modify or change Metabo Analyst as it exists. You would have to, it would just be a lot more tedious to be able to do that. If you did want to write your own applications or let's say a specific application to analyze metabolomic data in the way that you do, you could do that in R. But if you were to need to just use the functions that are already available in Metabo Analyst for doing what are out of the box metabolomic data analysis, that's something that Metabo Analyst already does well. So Metabo Analyst is a tool for metabolomic data analysis. R is, or Shiny is, a general framework to build any kind of application, including metabolomic data analysis. What is the typical time it takes to build a similar visualization app? Not very long, Emre. Depends upon how experienced you are, but I would imagine that the kind of application that Richa showed here, would take you anywhere from like a couple of days to a few days if you're a very beginner user. But if you're an advanced user, if you've built a few applications before, it's probably less than a day or a couple of days. Again, these the visualizations that you saw, the tables that you saw on the Shiny application itself, those are available as widgets or like just tools that you can pick up from the R standard library and put into your application. Same for the code that analyzes, let's say, RNA-seq data. Those are available as R packages. You can use them pretty much out of the box. The great thing about Shiny or R, the ecosystem, is that there are a lot of packages already available for doing many different kind of biological data analysis, and often new publications release an R package again, which can be used to do the kind of analysis that particular publication has. So it's a very, very rich ecosystem in that sense. Charu's question. Richard, do you want to answer that one?
Yeah, so Charu has questioned that whether we can do functional enrichment of the data sets and how can we do pathway analysis. So yes, Charu, the Shiny app that we have built here was only for visualization of the downstream of how the differential gene expressions are there. And for functional gene enrichment, you can always get the list and use Enricher and other tools or PathPanther for that. But yes, if you want to develop a Shining app to do that, it's doable because it would probably it's about searching the gene signature into a database, and that can be done in R. And if it can be done in R, it can be translated to a Shiny app. I hope that answers.
We have another question.
Ricardo is asking, the upstream analysis of RNA-seq mapping and quantification is made outside R or Shiny. I guess you can handle it. So essentially, Ricardo, we have workflows that does the upstream analysis. And one of the limitation of Shiny is that you have to modify it a bit to handle a large amount of data and to do a lot of analysis. But it can be done in an R Shiny app platform. And here we have at Elucidata itself, we have developed a workflow which would follow this entire workflow, and it's a Shiny app integrated to Poly platform. So we could do it here.
Done in different frameworks. The way, at least on Poly, you can integrate those different applications to a Shiny application. So take their output and feed it to a Shiny application. You can also do that more generally as well. There are many applications available in the community for analyzing RNA-seq data, and you can take the output and plug to Shiny. But yes, we do handle it. We have done it for some of our partners previously as well.
Okay, do we have any further questions?
Great. So we would be very keen to also learn. Ricardo says, yeah. I think integrating, if you want to integrate the upstream analysis of RNA-seq data with downstream analysis, Shiny is probably a good way to go. And you can take the output from upstream analysis and move that to a Shiny application, then do the downstream analysis. That's a pretty good way to go. Keith, what does it cost to use Poly, for example? We have different kind of plans for industrial customers. It's different from academic customers. It depends upon the amount of usage that you have. And we price it per workflow. So let's say if you had a workflow, we could develop it for you, and it's an annual subscription for 5 users within your lab that takes care of, let's say, developing a custom application, putting it on the cloud, and maintaining it as well. So do get in touch if you want to know more details. We have different plans for industrial customers and academic customers as well. I just wanted to add, while we're still here, that if you have requests for what we should do next, if there's a more hands-on tutorial of Shiny application building that would help, we're happy to host that. Or if there's something else, a specific kind of Shiny application, we're again happy to do that as well. So let us know in the comments here or on email, and we'll be happy to take them up in one of our future webinars. We saw that a lot of people have not built Shiny applications based on the survey that we just did. So maybe one of the follow-ups will be a more hands-on webinar about building a Shiny application as well. And do join us for that.
Okay.
Great. Let us know if you have more thoughts, questions. Thank you so much for being here.
Yeah. Thank you all for attending, and hope you visit us on our website.
Thank you.
Thank you.